Please join us in welcoming Professor Gordan Lauc, SCD, PhD, a global leader in the field of biochemistry and molecular biology. Prof. Lauc serves as professor at the University of Zagreb and directs the National Center of Scientific Excellence in Personalized Healthcare. He also holds honorary professorships at the University of Edinburgh and King's College London and is a distinguished member of the Johns Hopkins Society of Scholars. In 2017, he co-founded the Human Glycome Project, a groundbreaking initiative that explores the role of glycans in human health and disease. His team has pioneered advancements in high-throughput glycomic analysis, leading the way in applying glycan biomarkers to precision medicine. Through an integrative systems biology approach, they connect glycomic data with genetic, epigenetic, and biochemical insights to understand the physiological and pathological role of glycans. Professor Lauc's contributions to science are extensive, with over 200 published research articles cited more than 5,000 times. He has led or co-led numerous major international research projects, including several funded by the NIH, FP7, H2020, and ESI funds. Additionally, he is the founder of Genos, a biotech company recognized as the global leader in high-throughput glycomics. Please give a warm welcome to Prof. Gordan Lauc.
Good morning. It's great to be here again. I was here 2 years ago. It was a long trip from Europe. When I see you all here, I think it was worth it. So thank you, A4M, for inviting me here. And thank you, Jim, for your kind words at the opening session, advertising glycans and my talk. So I will try to introduce a very complex topic to you, the topic of protein glycosylation. I do hope that soon we'll have a full A4M training course in glycans. So please consider my presentation today to be kind of a tasting menu into the fantastic field of protein glycosylation. So what I will try to achieve today is to explain you some basic concepts of glycosylation, which is the most complex post-translational modification of proteins. Then I will try to present you how glycans are important as a mechanism which modulates and monitors inflammation at the molecular level. And finally, I will tell you something about interpreting changes in the composition of the IgG glycome in the context of disease prevention and personalization of anti-aging interventions. But before I move into glycans, I will remind you about the three problems we have in medicine today. And one of the big problems is that there is no standard human. We are not all the same, we are all different. But the majority of medical knowledge is based on the assumption that there is a standard human. If you talk about longevity, we know it's a balance of healthy sleep, healthy diet, moderate physical activity, and stress, which make us healthier and live longer. But on an individual level, for a given patient, it could be mainly dietary problem, or a sleep problem, or a stress problem, because we are not all the same. And this is one of the reasons why the only longevity intervention, which repeatedly works in all studies, is a caloric restriction. In the far left corner of my slide, it's a famous hungry monkey. It has been hungry for 20 years. 20 years of caloric restriction. All tests are showing him to be better and healthier than the normally fed monkeys, but if you look into his eyes, he's not happy. So caloric restriction is not a solution. We do not want to be hungry for the rest of our lives. But to find interventions which work is not easy. We know that today 80% of smart drugs are being wasted. We are giving them to patients which do not respond. But the same is the case for lifestyle intervention. 80% of all these difficult lifestyle interventions people do actually do not help them because they're doing the wrong thing. And we have no ways of knowing what is the right thing for our patient unless we have a good biomarker. So now we can just try and see whether it works. But eventually, we want to have a biomarkers which will tell us what will work for a given patient. And this brings us to problem 3. And this is that for the last 50 years, majority of science was focusing on genes. Technically, this was a great success. We can sequence our genes. A couple $100, we get all the letters in our genome. But we don't know what to do with that information. And even once we learn, it will not help us much. Because what we also know now is that 20% at maximum, and some newer studies say even 7 to 10% of longevity is genetics. So it's not our genes which determine how long and healthy life we are going to have. It's our lifestyle. And we do not have good biomarkers. So what I would like to propose to you, to go back to things which work, and these are glycans. Because the first biomarker which has been introduced in personalized medicine are actually glycans. Blood groups, ABO blood groups, are chemically glycan structures. And they're being used for over 100 years to stratify patients. But the majority of glycans are very complicated structures, multiple glycans per protein, and they're not easy to analyze. And until maybe 15 years ago, there were no methods which could help us analyze glycans in a large studies. And we need large studies to be able to confidently know whether something works or not. But in the last 15 years, we managed to raise over 40 million euros in research grants to develop glycan analysis technology. And today, my lab is analyzing over 80% of the glycom data generated in the world. And we don't do it in any people. We do it in collaboration with the best researchers all around the world, some of them even being in this room now. And so far, we analyzed over 200,000 people. And by combining high-end glycan analytics with the fantastic cohorts collected at Hopkins, Yale, Stanford, Cambridge, Oxford, and so on, we managed to generate a lot of high-quality data and publish it in high-quality journals. So we publish approximately 30 papers a year, many of them in the leading journals, including Circulation, Circulation Research, Gastroenterology, Nature Immunology, and so on. So most of the things I will show is our own data published in the high-quality, peer-reviewed journals. So let's go back to glycans. What you see here, revolving in the middle, is S-glycoprotein of the SARS-CoV-2 virus. Everything hairy on the surface is a glycan. Without the knowledge of glycans, we cannot understand that protein, but also other proteins. Majority of proteins are glycosylated. One of the famous examples is immunoglobulin G. Antibodies, our main weapon in fight against infections. It's a glycoprotein, and glycans determine which kind of effector functions will this immunoglobulin elicit. So with a specific type of a glycan, so-called G0 structures, it will be more pro-inflammatory, it will cause inflammation, it will kill the cell. But if you add sialic acid on top of a glycan, immunoglobulins will start suppressing inflammation. It will ignore the antigen. It will think the antigen is a friend. And for example, this is something what we did at the multiple COVID boosters. So antibodies of people who received multiple COVID boosters actually have a lot of sialic acid. They ignore the antigen. So analogy I like to make are these two birds. A regular bird with the feathers, that's a glycoprotein, protein with glycans, and a bird without a single feather. That's only polypeptide part or a gene. And we could be the best scientists in the world. We could have all the money in the world. But if we are trying to understand flight, and we use this bird as a model, this is the closest to flight we could come, the bird walking. Because the bird without its feathers cannot fly. The protein without its glycans does not function normally. So to understand biology, to understand what is going on, we have to look into glycans. And the majority of proteins are glycosylated. But the word of caution, unfortunately, there is the word glycation, which is something completely different. So glycation is a random reaction between glucose and the proteins. We quantify it as HbA1c or A1c or advanced glycation end products. It's a random damage. It's like taking a shotgun and firing at a protein. Glycosylation is a sophisticated modification which makes proteins better. Actually, The glycosylation is an additional layer of complexity between gene sequence and a protein function. We have only 20,000 genes. Simple bacteria has 5,000 genes. We are not only 4 times more complicated than E. coli. We are millions of times more complicated than E. coli. And this is achieved by having a glycosylation which is produced through interactions of hundreds of genes. So it's a kind of a network information in genetic polymorphisms, in epigenetic regulation, in the metabolites interacting with the pathway, deciding which glycan we are going to add to the protein. So the final protein will have a genetic component, fixed for our lifetime, fixed for generations, and a glycan component which we can actually change. So from a functional point of view, alternative glycosylation, adding a different glycan to a given glycoprotein, is analogous to coding mutation. It's changing the structure of a protein. It can literally kill you. But from informational or evolutionary aspect, glycan composition is inherited as a complex trait. And this enables diversity without the risk to our precious genetic heritage. We demonstrated this very elegantly in a study of a collaborative cross-cohort of mice generated in Western Australia, where the Grant Morahan took 8 founder strains, as diverse as he can find them, crossed them for 3 generations, and then inbred them into strains. So more or less, he generated strains of cousins, as similar to the clones of cousins as possible. And when we looked into protein glycosylation, some of these strains were completely different from the rest of mice. So mice can become different just by reshuffling allelic variants through 3 generations. And we see this also in humans. So when we look into people, into glycans of people, we don't see them as all the same. What you see on the right part of this panel is IgG glycan composition, different color, different glycan. So people are different in the way they glycosylate protein. And again, glycation is something completely different. I'm talking about glycosylation. So glycans are the ultimate layer of molecular complexity. They integrate genetic, epigenetic, and environmental factors. So billions of years of evolution, everything what happened to us in our lifetime, what we think, what we eat, what microbes we have in us at the moment, integrate into chemical structures, which do many different things, among other things, regulate low-grade systemic inflammation. And this is changing through our lifetime. So among our 200,000 people, there are people of different age, younger and older. So when we looked in the different cohorts, and this is China, Croatia, UK, Estonia, people are changing through age. Blue are men, they change more or less linearly. Red are women. They are protected from aging in the reproductive period, then have a rapid acceleration, and then kind of catch up with men. So when we published this over 10 years ago, the editors of Gastroenterology published an editorial asking, are glycans the holy grail of biomarkers and aging? And I believe they are, and I hope at least some of you will believe after my presentation. So when we looked how glycans change with age, we see that people are becoming older in their glycome, but the pace of aging is different. And what determines this pace of aging is largely lifestyle. So people who have accelerated glycan aging will have high insulin, high fibrinogen, high HbA1c, high BMI, everything pointing to the unhealthy lifestyle. And we use this knowledge to develop something called the Glycan age test, which is today commercially available in over 1,000 clinics all around the world, which is quantifying IgG glycans and mapping them on the network of 200,000 people we have so far, and kind of putting you on a trajectory of accelerated or slower aging. And of course, I am conflicted here. I am inventor of the test. I'm co-founder of a company. So you don't have to take my word for it. Look at the data and make a decision for yourself. But the advantage of me being part of the company is that I can offer discounts. So if you come to the booth 8080, you can get the Glycan age test with a 25% discount. But let's go back to science. So it's always good to have independent verification of what you do. And this is a study done in Qatar at the Weill Cornell. So they purchased more or less any molecular marker which can be purchased. Epigenetics, proteomics, several types, lipidomics, metabolomics, glycomics. And when they looked into molecules which associate with aging, 9 out of the 20 strongest associations were immunoglobulin glycans. So glycans change a lot with age. And there was a recent perspective paper published by the Consortium for the Biomarkers of Aging, which we are not part of, which clearly said that glycans are the only commercially used test to track individual response to lifestyle challenge. And the reason why glycans are the only test which is commercially being used to monitor interventions is that it's the only test which has analytical accuracy that enables monitoring of an individual patient. So this is my personal IgG glycan composition through approximately 2 years. Each time point is a sample sent to the lab, results got back, another sample, so it's not analyzed as a batch. It's a series of results. And you can see they mostly do not change unless something happens. And we can see in the middle, there is a decrease in the glycan, which is kind of protective of a heart attack and stroke, and an increase in the glycan, which is monitoring, which is correlating with inflammation. And I knew it was bad, so I tried to find out what is going on. And I found a cryptic root canal infection. I fixed it, and these biomarkers went back again. And to be able to monitor individual patient, we need analytics which is stable enough. For example, if we would do a similar thing using any kind of epigenetic test, they would just go up and down simply because of an analytical variation. So the key problem with epigenetic tests is that they are great for cohort work. You can do a lot of research, you have hundreds of people of one or the other type, and then you compare them. But to take an individual patient, the analytical inaccuracy is simply too large. This is a study we are doing together with Jamie Hayward and Alden Scientific. They are quantifying everything what can be quantified in a cohort of people. This is Jamie himself, and an epigenetic test done repeatedly in a short period of time. So his epigenetic age went up 5 years up, 5 years down in 2 days. It's not biological, it's just analytical. So until there is a significant improvement in analytical technology used for epigenetic testing, the only test we can actually use to monitor individuals are glycans. Again, this is me a few years ago when there was a rapid acceleration of my glycan age, and this was COVID. Actually, during COVID, I had a pretty severe COVID. I gained 4 years, which I never got back. So some people went down after COVID. Some people just stayed on an elevated age. And the second important question is, does it mean anything? So when you measure some kind of biomarker, what does it tell you? And the difference between glycans and other types of biomarkers is that glycans are actually functional effectors which regulate inflammation. And if you change them, you know that something will change in an organism. This is a study did with Sarah Haag on a Karolinska, still not published, we should publish it soon, showing that glycans are actually predicting mortality, predicting dementia. Something we did publish was on a cohort level, comparing 27 different populations from all around the world, and people living in an area with a shorter expected lifespan had their glycans moving much faster to the older age than people who live in the richer areas with a longer expected lifespan. During the pandemics, we learned that glycan age is something which is a better predictor of severe COVID, and by the way, any other severe viral infection, than age itself. Also, in infected people, rapid acceleration in glycan age was the predictor of fatal outcome. So what you can see on the right-hand panel, you can see people accelerating, and then there's no further stops, further point, they died. So this rapid acceleration within a patient, within a short period of disease, was predictor of a fatal outcome. And in our 200,000 people, we studied dozens of diseases. Some of them have a similar pattern of glycan changes, some of them have different. There's excellent 2 review articles. Have a look if you want to learn more about glycans. But for topic of today, and what is aging, is that in majority of diseases, we see glycans changing in a way of accelerated aging. So a healthy person will look younger than a person of the same chronological age with some of the diseases. And the changes in glycans happen up to 10 years before the disease is actually diagnosed. You have multiple large studies showing changes of glycans visible 7-10 years before first diagnosis. And this is because we are quantifying low-grade systemic inflammation. Inflammation is contributing to the disease development. And this is a textbook knowledge already. So there are excellent reviews going 10 years back in Nature Immunology, immunological reviews showing that glycans affect multiple molecular levels of inflammation. And this has consequences. For example, when we looked into large cohort of 3,500 people in UK, we saw that glycans strongly correlate with the cardiovascular disease risk score, so the risk of having heart attack and stroke. Then we looked into the prospective cohort of 27,000 people collected 30 years ago, clinically followed up. We know who had a heart attack and stroke, and glycans were as predictive as the entire AHA score. And interestingly, different glycans predict heart attack and stroke in men and women. Published in Diabetes Care, a good paper, but one paper is never a proof. We did a replication cohort, again, different glycans, men and women, very similar hazard ratios. And then we did additional 2 replications published in Circulation Research a couple of months ago. So we are pretty confident glycans predict heart attack and stroke. And there are some molecular mechanisms which could explain it. We know that glycans affect endothelial biology, and you can actually hack it. This is just a mouse experiment, but if you feed mice a supplement which improves IgG cellulation, it actually protects obese mice from developing hypertension. Another very important aspect of glycan research is diabetes. So this is a cohort of 2,000 twins measured in 3 time points. And we see that glycans change as somebody is moving from a healthy state to insulin resistance. And it's a continuous progress through 10 years. You can kind of monitor the track of going to insulin resistance. Of course, this is not a perfect prediction, but diabetes is not a disease, it's a symptom. There are multiple mechanisms. And we even found one where glycans are perfectly predictive. So there is one pathway to diabetes, which is more or less regulated by glycans. And interestingly, these same glycans also predict the lipemic and glycemic response after a meal. So it's relevant for kind of a food metabolism. And these same glycans can be converted back to the healthy state by beneficial lifestyle interventions. And I think this is the key question. Can we revert these glycans? Can you make a decision and do something and make us healthier? So we did a number of studies. We published dozens of papers showing that physical activity, dietary intervention, sex hormones, anti-inflammatory biologicals, they can revert the glycans. I cannot show you everything here. I can show you, of course, the famous hungry monkey. So we had 1,000 people on 800 calories for 2 months. They improved, but nobody wants to stay at 800 calories. So we then put them on 5 different maintenance diets. And on each of the diets, some people got better, some people got worse. So there is no magic diet. There's no single diet which works for everybody, because we are not the same. And this is probably the reason why we have so many different books about a healthy diet. And people are so passionate about what they think it works. And it probably works for them and for some people, but not for everybody. What is good is that if you start losing weight anytime in your life, your glycan age will improve in the 30s, in the 40s, in the 50s. So it's never too late to start living healthier. Physical activity also has positive effects on glycans. People who exercise regularly look younger. But professional athletes actually turn out older. So too much physical activity is not good. And if you take a regular couch potato and put them in an exercise program, most of them will get worse for at least 6 months. Only after 9 months of regular physical activity, people actually get better. And these things should really be personalized, because what we learned, the majority of trainers simply overtrain their clients. But we would all like to have a magic pill. We don't like to exercise, we don't like to be on a diet, we want to take a pill and have our unhealthy lifestyle. So one thing we can do, we can take anti-inflammatory biologicals, they work. Side effect is cancer. So I don't think this is the way we should do it. Hormones work. So if there is a deficiency of estrogen, estrogen supplementation works. This was a very elegant study where the menopause was chemically induced. Supplementation with estrogen completely rescued the signals. So women who were depleted of estrogen, aged 9 years in the 6 months. Estradiol supplementation prevented it. And when we look at the population level, I showed at the beginning, we see this rapid acceleration in the perimenopause period. And actually what we have seen is that acceleration in glycan aging is one of the first symptoms of perimenopause, because glycans are the strong influence of estrogen, and estrogen is very hard to quantify because it really goes rapidly up and down in the perimenopause period. And IgG glycans actually integrate a couple of weeks of estrogen concentration. So what we believe is actually that IgG glycan composition is a kind of HbA1c for estrogen. Something like HbA1c for glucose, that's IgG glycan for estrogen. And we do have some clinics who are kind of optimizing HRT based on IgG glycan composition. Maybe that's not the magic solution. It helps when there is a hormone deficiency, but we would like to find the points where we can kind of druggable points below the estrogen. We are trying to find it. Interestingly, plasma exchange works. There was a presentation yesterday, one today in the afternoon about the therapeutic plasma exchange. It does improve IgG glycan composition, but it does take 6 months. It's not very rapid. Exosomes, some exosomes are magic. Exosomes from newborn pigs will make rats live 50% longer and reduce epigenetic and glycan age. Metformin, many people take metformin, but is metformin a magic anti-aging drug? I would guess no for healthy people. In our placebo-controlled randomized trial, only a single individual in the entire trial actually benefited. But of course, if you're diabetic, it helps. But not everybody. The same for GLP-1, same for SGLT. Microbiome, extremely important. Microbes and glycans have continuous crosstalk. And I think that glycans can be used to monitor this path to the disease. And we do have people monitoring their glycans, seeing something going wrong, improving, getting better, then getting worse again and getting better again, depending on what they do. And at the end, I have to acknowledge there is a large team of people working on this. We have our research labs, both the glycan and epigenetic team, and we have a glycan age who is commercializing this part. And I cannot take any questions here, but you can come to the booth 8080. I'll be there most of the day today and tomorrow, and happy to talk to any of you. And thank you for your attention.